Happy New Year to all of you. And we thought together with Roche that this was a very good way to start the new year, to have the Generation HD2 trial presented in more depth and discussed among us, and the importance of this trial. So I'm really happy for that collaboration with Roche and for the opportunity to do this. And for you all to be here, really, that's really appreciated that you choose to be here and listening together with us. And of course, an extra warm welcome to our dear presenter, Sarah Tabrizi, Professor Sarah Tabrizi. Really nice to have you here. It's an honor.
Thank you so much, Astrid, and Happy New Year to you all. And I hope this year is going to bring us good news for Huntington's disease. And so to start the new year, I thought I would give you an update on Generation HD2 and the rationale behind the study. And so thank you very much for allowing me to do this. So I'm just going to share my screen. So first of all, I'd like to do my disclosures through my university. I have undertaken consultancy services, and I'm on scientific advisory boards for Alnylam, Annexon, Atalanta Design, Roche, Iris, Lattice Bio, Life Edit, Novartis, Pfizer, PTC, Regenta, Takeda, Unicure, and Vertex. Importantly, all the payments for these consultancies go through my university, and it goes to both UCL, and what doesn't go to UCL is contributed towards my research in the Huntington Center. So today I'm going to give you an overview of the tominersen program. I'm going to give you some key findings from Generation HD1. I'm going to go over the new study and give you an update on the new study. So the overview of the tominersen program. So tominersen is an investigational molecule called an antisense oligonucleotide, which is basically chemically modified DNA that has been designed to bind to the Huntington message. Through that binding, the antisense oligonucleotide degrades the Huntington message. Then that results in less of the Huntington protein being made. So that's how the antisense oligonucleotide works. So this program has had a very long program history. Program actually started in 2005 at Ionis Pharmaceuticals. And then in 2013, Roche and Ionis had formed a partnership, which was a Huntington's disease partnership. And between 2015 and 2017, there was the original study, which I led sponsored by Ionis. And that was the first study in Huntington's disease to show that you could lower in the CSF mutant Huntington. And Roche then licensed this ASO, which is called tominersen, to take forward for clinical development. Between 2018 and 2021, there was a number of studies, an open label extension of the original phase 1 study. There was a natural history study, which is subjects who were enrolled but got no drug. Then there was a phase 3 study, which I'm going to come on and talk about, called Generation HD1. And there was 2 other studies, a Gen Peak, which was to try and understand more about the molecule, and then Gen Extend, which was an open label extension. In 2021, on the 21st of March, the trial was halted dosing in the Generation HD1 study. And data analysis was conducted to evaluate if there was a path forward or a new hypothesis to test. And now the scientific study, which is a phase 2 study, which is a safety, tolerability, and dose finding study, is now ongoing. And this is called Generation HD2. It's well underway globally, and 45% of the participants are recruited. So the key findings from Generation HD1. So the subjects in Generation HD1 showed a consistent pattern of clinical progression over time in key outcome measures. This was one of the largest studies that had been done in Huntington's disease, and it gave interpretable study data, i.e. that meant that you could make conclusions about the data to enable recommendations by the Independent Data Monitoring Committee. So an independent data monitoring committee is set up for all trials, and they are independent from Roche. They're independent from the investigators, and they are there to monitor unblinded data and raise any concerns. And based on an overall benefit risk assessment of the Generation HD1 data at 17 months, the IDMC recommended to stop dosing in all the study arms. And many of us remember that, and it was devastating at the time. So the 120 milligrams, so what led to this? The 120 milligrams every 8 weeks. In the 120 milligrams given every 8 weeks, tominersen had an unfavorable pattern compared to placebo. The subjects getting the drug frequently were doing worse than placebo. The 120 milligrams every 16 weeks was less clear. The safety profile appeared comparable to placebo. However, in the 17 months they were tested, there was no apparent benefit observed with the CSF mutant huntingtin lowering. So in Generation HD1, the adverse event profile of the 120 milligram dose given every 4 months was actually comparable to the placebo group. And you can see here in orange is the 4 monthly dose of 120 milligrams. And this is the placebo. And you can see here, it actually looks pretty similar overall in terms of, and actually there were less SAEs or severe SAEs in the 16 weekly group than the placebo. So the AE profile was similar. This is the data, which is the change from baseline to week 69 for the total functional capacity. So here, I'm just going to talk you through what the colors are. 8 weekly is blue, 16 weekly is orange, and gray is the placebo. This is the total functional capacity, which many of you know about from coming to clinic. And going upwards is improved outcome. And at week 69, which was 17 months, the 8 weekly group of 120 milligrams were doing worse than the gray group, which was placebo, and the 16 week, there was no statistical difference. In a post hoc analysis, one of the questions Roche had was when they had this data, was there anything in the data which would suggest that people who were younger with less disease burden, i.e. earlier in the course of the disease, had anything to suggest that the total functional capacity was going in the right direction? So the statistical team divided the population into those with low exposure, i.e. the 4 monthly dose at 120 milligrams, those who were aged under 48, and the CAP score was a measure of their disease burden. So they had to have a lower CAP score versus the higher exposure aged over 48 with a higher CAP score. And what, on a number of measures, but I'm showing you total functional capacity, the low exposure group went in the improved outcome direction on all of the clinical measures that were tested. So what does this mean? And I'll come on and talk to you about that. Now, many people are interested in what happens to neurofilament and to ventricular volume. And the statisticians and the pharmacologists at Roche pooled all the data across the 8-weekly group and the 16-weekly group with the loading dose. So in this study, the subjects, when they were recruited to Generation HD1, had 2 loading doses, 4 weeks apart, of 120 milligrams. Then they were enrolled into either 8-weekly or 16-weekly. And we have all learned a huge amount about the exposure and the loading dose. And I'm going to come on and talk to you about that. But when we take the time after first dose, this is quite a complicated graph, and the change from baseline. This is the placebo. This is CSF neurofilament in red. This is CSF mutant Huntington in pink. And this is ventricular volume. Now, ventricles go up as part of disease progression. CSF neurofilament also goes up as part of disease progression. At the highest doses, as you know, there was an increase in neurofilament and an increase in ventricles. And you can see here, if you can see my pointer, it is dose dependent. So, and the ventricle increase and the NFL increase are all dependent on exposure to the drug, which is dose. The lowest exposure quartile, and this is still at 120 milligrams, but those in the 16-week dose had CSF mutant Huntington lowering, but the ventricle increase was actually very similar to the placebo, and the NFL rise was mild compared to the higher doses. And actually it was very subtle. So there were smaller ventricular volume changes at lower exposure. And this lower exposure quartile is because it's divided from 4, is targeted in our new study. So what other information has become available? And this is data that actually is not published, but has been presented. I've talked about neurofilament. It's an important biomarker. I did a recent HD Youth Organization webinar about neurofilament and what it means. And neurofilament is a marker in the brain. It goes up with disease progression, and we're very interested in it as both a safety biomarker and also a biomarker of potentially saving nerve cells or neurons. In the 120 milligram 8-weekly group in blue, the NFL was above the placebo, which is here at the dotted line at 0. And here is the difference in percentage change from baseline. The orange is the 120 milligrams every 16 weeks. And when it was looked in plasma NFL at day 101, the orange fell below placebo at all the time points. You can see here in orange. Now, this is potentially interesting. It potentially gives us a new hypothesis to test, which is what I'm going to come on to with Generation HD2, because this drop in NFL may suggest that in the 16-weekly group, that there may be some potential saving of neurons. There was also a digital motor score in the study, and these were tested balance, chorea, drawing a shape, speeded tapping, a 2-minute walk, and doing a U-turn. And all of this data was collected, and it was amazing the number of people who were able to complete the study. And I think digital motor testing is important for clinical trials because it's done regularly rather than when you go to the doctor and see the neurologist at the clinic. And accounting for the differential dropout, the digital motor scores showed a trend in favor of the 120 milligrams every 16 weeks. This is improved outcome. And you can see here, this is the orange is the 16-week, gray is the placebo, and they are clear, they look different. It appears that the 16-week on the digital motor is going in the improved outcome direction. Now, this is a subgroup analysis. This is a post hoc analysis. But what does it tell us? It tells us, it gives us a hypothesis, because science is all about testing a hypothesis. These data, the younger subjects with less disease burden, the neurofilament, the digital motors score, all give us a new hypothesis to test. And the new hypothesis being tested is the reason for the new study. And I'm going to tell you about that. So we know that tominersen gets into the brain regions vulnerable in Huntington's disease, including the cortex, striatum, and thalamus. And this was presented by Blair Levitt at the CHDI conference in April last year, where a subject had come to post-mortem in one of the studies, and it showed that that tominersen got into all the brain regions. The drug currently is the only one that has been shown to lower mutant huntingtin in CSF. There is no other drug that to date has publicly shown data that they can lower CSF mutant huntingtin, which is coming from the brain. So tominersen, because of that, deserves to be tested in this new study. The adverse event profile for the 120 milligram dose was comparable to the placebo, and the same is anticipated at even lower doses. And based on the post hoc analysis, the hypothesis being tested is, if we have much lower doses, is it safe, tolerated, and does it show any evidence that it may slow disease progression in younger adults earlier in the course of the disease? And the neurofilament and ventricular volumes are something that are being monitored very, very regularly in the new study. But it appears that lower exposure may prevent those increases from the previous data. Now, there's also some other new data about neurofilament that I think is important. The subjects in the natural history study, they were just regular subjects, they rolled over into the open label extension called Generation Extend. But those subjects were not given the loading dose. And in those subjects, even though they got 120 milligrams every 4 months, they had no loading and there was no change in their neurofilament. So the new study is testing a new hypothesis. And a hypothesis is when you have data that then helps you generate a reason to do a new study to test if lower doses of tominersen are safe, tolerated, have target engagement with mutant huntingtin in CSF, and if there are any signals that it may be of benefit. Again, this is the only drug that has to date shown that it lowers CSF mutant huntingtin. So the exploratory post hoc findings showed that younger adults with symptomatic Huntington's disease, but earlier in the course of the disease, who received lower tominersen exposures may have clinical outcomes that are going in the right direction. It's not statistically significant, What a post-hoc analysis does is it generates a new hypothesis. The new study is focusing on adults with prodromal, which is very early subtle symptoms, or early symptomatic Huntington's. The doses being tested are much lower. It's lower and less frequent dosing, and importantly, there is no loading dose. And Generation HD2 is looking at safety, biomarkers, and any evidence of clinical efficacy trends. It's not powered to tell us if it's slowing disease progression. It's just going to tell if there are any trends. So this is the design of the study. And one thing I can tell you, and I don't work for Roche, I think that the community, but Roche in particular, and the community have, this was the first trial of a Huntington lowering agent. In drug development, it is rarely the first trial that is the success. It's a slow, tough process. But what the study did tell us was a lot about Huntington's disease, progression, lowering Huntington, antisense oligonucleotides, and dosing. And I think Roche have really learned a lot about the dosing regime. And that's one of the reasons they dropped doing the loading dose, because that was not the right original thing to do. There was a good scientific rationale at the time, but this is how we learn from science. And you have to do the scientific experiment to try and understand. So the new study is a smaller study in early prodromal and early manifest Huntington's disease versus placebo. It's got 2 dose levels with no loading. It's testing 100 milligrams every 4 months. And so even lower than the original studies, 60 milligrams every 4 months, which is half the dose, versus placebo. There is an independent data monitoring committee who is regularly reviewing unblinded data, and they are making then recommendations that the study can continue. So what this study is a phase 2 dose finding, safety, tolerability, and biomarker study. So what's the key differences in the new study compared to the old one? I've already said there's no loading. This is the old study. You can see there was quite a lot of the drug being administered. This is the new study, much less drug, no loading. So it's much lower doses. It's a reduced dosing frequency, only every 4 months. There's no loading dose, and the CSF is being sampled between dosing visits to really try and understand target engagement. So the study is now underway in 15 countries, 70 sites have activated, and over 45% of participants are enrolled. And you can see here the European sites. As of the 12th of September, the Independent Data and Monitoring Committee had reviewed unblinded data from 50 people, about 50 people with 1 to 2 doses, and recommended to continue the study with no changes. So that's the study now. It's still recruiting. And I'd like to thank you, the HD community, for your ongoing collaboration. I'd like to thank all the study participants, their families, the investigators and site staff. And I'm one of a group of people on the Tominersen Steering Committee. I think importantly in science is we have to test new scientific hypotheses. This is being done in a smaller group of patients earlier in the course of disease compared to the subjects in Generation HD1 with much, much lower doses and exposure. And we are all going to be working together to find out if tominersen has any potential effects at the doses given in this study, and also carefully monitoring safety. So I'd like to thank you for listening, and I'm very happy to take any questions. And Peter McColgan from Roche is also here and is also happy to take any questions. So I'll stop sharing. I'll reshare again if anyone has any questions that I need to go back to the graphs, but for now I'll stop sharing.
Thank you so much, Sarah, for a really clear and good presentation. And you were also able to speak quite slowly, which is really good for me, not being that fluent.
I hope it wasn't too fast for anyone, because I was trying to get across quite complicated ideas.
Yes. No, thank you so much for that excellent presentation. And we have a couple of comments in the chat, and feel free to type down whatever your questions or concerns or comments.
I'm happy to answer anything that people want to ask about the new study. Yes, and yes. Peter will answer on behalf of Roche.
Yes. So Dina, please.
Yes, there's actually only two comments in the chat. So the very first question is, I'm curious about the new Roche Ionis partnership for HD other than tominersen.
Yeah, I think that'll have to go for Peter.
That's not really about Generation HD2, but anyway.
It's a great question. I know that there has been a press release went out, I think, a number of months before Christmas. There's not much we can say about that at the minute, but when I'm able to share more information about that, I'd be delighted to share it with the community.
And the second question was, why can't prior participants in the tominersen trial be in the trial even as a subset group?
Peter?
Yeah, I'm happy to take this. So just to clarify, if people had been on placebo in Generation HD1, then they can undergo screening for Generation HD2. So the question about people that had tominersen treatment on the previous studies, the challenge is that if people had previous treatment, then it's really difficult to understand the lower doses that we're investigating in Generation HD2, and it would make the data very difficult to interpret. It doesn't mean that we aren't continually looking for opportunities for those that were involved in the previous studies. We acknowledge the great amount of dedication and commitment those individuals went through for the previous studies. So we're always looking for opportunities to enable them to be involved. I think the first step is for us to replicate what we've seen in the post hoc analysis in this smaller subgroup of individuals who are younger with less advanced disease. But if we can do that, the hope then is that we can expand this. And our ultimate aim is to try and treat as many people as possible of a wider range group and wider CAP scores and disease burden scores. But I think the first thing we have to do is replicate the signal that we've seen in the post hoc analysis.
So another question, will tominersen be tested with a new stage system?
So I can answer that, and Peter can answer that if appropriate. So the first thing is to do this study, and this study is really in people with late HD integrated staging system stage 2 and early HD ISS stage 3. This study is all about, is there a dose that engages the target mutant huntingtin? Is it safe? Is it well tolerated? And are there any signals that are supportive of continued development? If that happens, the program will continue, and then the program may expand to test those in much earlier stages of disease. But at this stage, we can't do that until we've tested its safety, its tolerability, and also its profile in people in HDSS later stage 2 and 3. I am as passionate as many of you to eventually do trials in stage 0 and 1. the big thing that we have at the moment is that we don't yet have ways of measuring progression in stage 0 and 1 that could be used in a trial. Now, I think NFL for stage 1 and brain imaging is and digital motor testing will become very important for stage 1 and possibly in stage 0. But that work is ongoing to establish potential measures and endpoints for those trials. So I hope in the next few years that we will be doing stage 0 and 1 trials. Tominersen may be part of that if this program, this new Generation HD2, looks favorable. I don't know if you want to add anything, Peter.
Yeah, to come back to the idea that we need to replicate the signal, I think with Generation HD1, we used the previous system because the new staging system wasn't developed at that time. So if we want to replicate the signal, we need to use the old system to have the same participants with the same CAP scores and the same age. But as Sarah said, if we can replicate that signal, then we would like to ideally test all stages with the caveats that we need sensitive biomarkers for people that are much earlier.
So I'm going to read this question because I don't understand the question, but maybe you, Sarah and Peter, will. What are beyond CAP score and CAG repeats the in and inclusion criteria in the new study?
Yeah, so I'll start. Peter, you can add. So the inclusion exclusion criteria of the new study is the inclusion criteria are obviously, as we've mentioned, is the CAP score and the stage of disease, which is prodromal or early symptomatic. Also, other inclusion criteria include stable mood, able to have an MRI, able to have a lumbar puncture, able to come to the study site. So it's for people with prodromal or early symptomatic. And it's the usual inclusion criteria, and able to have an MRI and lumbar puncture and ability to do the study and not to have active depression or mood problems. That's not an exclusion. They just need to be stably treated. Exclusion is if you are out with those criteria, but also if you're not able to have a lumbar puncture for any reason or not able to have an MRI, or have a significant mood problem. The significant mood problem is not an absolute exclusion because it can, as we know, treatment of the mood problem can then stabilize symptoms and subjects can then potentially be evaluated. The study is still recruiting, it's ongoing. So if you are involved at a study site that is doing the trial, then and you're suitable and you think that you may be suitable, you should ask your local doctor about the possibility of being involved. Peter, did you want to add anything?
I would just reiterate that if someone's interested in the study, the easiest thing to do is speak to your neurologist or physician. Then they can work with you and go through the inclusion and exclusion criteria to see if you can be involved.
And then we have how clinical meaningful are the changes observed in the post hoc analysis?
So the post hoc analysis data is going in the right direction. So clinical meaningfulness, we have the term clinical meaningfulness means how much that the change in the total functional capacity or the total motor score or the simple digit modality test, what change is likely, or in the composite UHDRS, all of which went in the right direction. Clinical meaningfulness is, just so people understand the question, is what change is going to impact a patient. So a total functional capacity score, slowing of say 1.5 points or 1 point over 1 year is clinically meaningful to a patient because it is slower disease progression. Total motor score over 1 year or 2 years may be 3 points or 5 points over 2 years. So those are used importantly in phase 3 trials because you want to have endpoints that are statistically significant from placebo, but you also want endpoints that are going to be clinically meaningful to a patient, i.e. in function, in motor scores. Now, the post hoc analysis is data that is going in the right direction. That's what a post hoc analysis is. And it doesn't tell you that the drug is going to work in that population, but what it does is generates a hypothesis that if those subjects who are younger, who are earlier in disease progression, can we see if that post hoc analysis of potential clinical improvement is replicated in a new study at lower doses in younger patients. So I would say the post hoc analysis as they stand are not clinically meaningful if you stick with the strict criteria, because clinical meaningfulness is an endpoint, particularly in efficacy trials. However, they are all going in the right direction, and we don't have anything in Huntington's disease that has shown clinically meaningful change in an efficacy marker like total functional capacity or total motor score, a disease-modifying agent that has changed the course of the disease yet. So it's the data shows that it's going in the right direction. The NFL and the digital motor data are supportive, and that merits the new study. So very long answer, but it's quite a complex question in terms of clinical meaningfulness. Peter, do you want to add anything?
Yeah, thanks, Sarah. I completely agree with what you've said. Just to add that when we look at the low age, low CAP subgroup, over a period of 18 months, if we consider the total functional capacity scale, we can see that it stays near baseline for those people that had Q16 120 milligrams, where it drops in placebo. And over that 18-month period, it drops by around 1 point. So the way I think about this is, in a participant who has early manifest, they may be in full-time employment, and a 1 point drop in TFC may be then they need to take a reduced work role, or they may stop employment. So in terms of what it means in the real world, I think maybe 1 point is the difference between you being in your employment and not being in employment over that 18-month period. So potentially that is how you could slow down the change over that 18 months. I really need to be, it's a post hoc analysis, so this is really a signal. And this is why we're doing the new study, to ensure that we can replicate the signal, to be confident that it's real.
And I think coming to clinical meaningfulness, exactly as Peter said, a total functional capacity change of 1 point over 1 year, we think is clinically meaningful because it is typically staying in your original job. Agree with everything Peter said.
So the next question is for you, Sarah, and it's from a woman named Sonja. She's asking if she could ask you a question in Farsi. So
I saw that. So although my father was Iranian when he was alive, Sonia, I don't speak Farsi anymore, unfortunately. I spoke it as a child and I've lost it. So I wouldn't be able to understand your question in Farsi. If there's anyone else who's a good Farsi English speaker on the call, I'm very happy to ask them to interpret for me and answer your question. But my Farsi would never be good enough to be able to understand your question. I'm kind of, it's just not good enough. Sorry.
Thank you. This is I will to talk because my English is not so good. I'm sorry.
Yes.
I am sick. I have this Huntington from my mother. I have after living in Iran, and now I'm living in Austria. And I have this sick, and I have passed by the doctor, and the doctor said to me, "You have 36% for Huntington," and I don't know what I did now.
Yeah, so I would have to, I think this is a clinical question, and I think you have to ask your doctor in Austria
Yes.
what he thinks about your symptoms, because it's difficult for me over a video to tell you about what stage you could have, but you can ask your doctor. And Austria has many good Huntington's disease doctors. You can ask your doctor to tell you about your stage, and then you can ask your doctor if there are any suitable trials. I think this trial, Generation HD2, is a study that may be expanded to other countries where Farsi in the future, where Farsi is the first language. But I think at the moment, to do the study, you would need to be fluent in another language other than Farsi for some of the tests. So that means you still can get involved in studies, but I think you should ask your doctor what stage of disease, and also as you learn the language better of either English or German, then you will become suitable for studies, if that makes sense. At the moment, there aren't any studies running in Iran, and it's something I've written to a lot. And we're hoping we can train a new doctor, and one day trials will be run in Iran if possible.
Yes.
And I have to apologize to Assange because I just assumed it was a woman's name, and apologies on my part for that. So the next question is, why are digital biomarkers not used in the new trial?
That's a question for Peter.
Thanks. We designed the new trial with in mind that we were looking at individuals who were younger with less advanced disease, and a lot of the feedback that we got from the community was these participants are going to be in full-time employment. They may have childcare responsibilities, really busy lives. So we wanted to keep to a minimum really the assessments in the study to maximize the flexibility and collect only the data that we thought was completely essential to answer the questions that we had. It doesn't mean that going forward, once and if we can replicate the signal that we've seen in future studies, we would use things like the HD digital motor score that Sarah shared today.
Yeah.
And there's two more comments on here, but am I allowed to ask a question myself?
Yes, of course, Dina.
Sure.
So why, because it's something you brought up, Sarah, why is the no loading dose important?
Because the loading, what we know from all the data so far is that the loading, and the reason Roche wanted to do the loading is the pharmacologists and the people who do the design of the studies and monitor how the drug may get into different parts of the tissues and how it interacts with Huntington. It was always thought with antisense oligonucleotides as a class of drug that if you gave loading doses to try and get as much drug into people as possible in a short period of time, it then would lower the level of your target more quickly, and then it would then reach what's called steady state quicker. So the reason that was why loading was done, and this was for all antisense oligonucleotides. This was not unique. But what we know from the dosing now is those two doses 4 weeks apart at 120 milligrams. We know from the no loading in the natural history study is that loading and the high exposure at that time contributed to the neurofilament going up. And so now with no loading, as was seen in the natural history study cohort who rolled into open label, they had no NFL rise at all and they didn't have loading. So that's why the loading has been dropped. And actually, Generation HD1 has influenced quite a lot of studies that are testing RNA therapeutics, because loading in these sorts of drugs in general, not just for Huntington's, is not a good idea. So Generation HD1 and the study has influenced a lot of other studies as well, because it's all about a learning in drug development.
Okay, then 3 more comments, as you can see on here, are people asking about why are trials not in specific countries or areas, Arab countries, Latin America, Sweden.
I saw that. So, I agree. Thanks, Dina. So, Peter, there's questions about why is there not any clinical trials in Arab or Emirates, Turkey, Sweden, Latin America, Venezuela, and Brazil. So,
yeah, I can.
You can see them.
Yeah. So when we look where to do trials, the sites that do the trials have to be able to have the infrastructure and the different things like MRI machines, the capability to do lumbar punctures and things like this. So that is some way in terms of how we select sites, but we're always open to looking at sites that do have the capabilities in different countries. So this is something that we continually look at, and if we move on to do a bigger study, we can look at having more sites in more countries as well.
And actually, I have a question for Roche. In your other programs, not Huntington's, but say in other diseases that do need an MRI and lumbar puncture, for example, are you able to do you do trials in Latin America and Turkey and they've mentioned Sweden and in Middle East?
It's a good question, Sarah. I only work on Huntington's disease, so
okay. Because I'm just in, because I think, you know, it's a very good question, but I think Peter's answer's right. Is if this trial moves forward to a phase 3 or and bigger study, for example, then more countries potentially can be taken on board. I think one of the things that those of us who work in Huntington's particularly feel passionately about is whatever treatment is developed, we have to make sure whatever treatment in the end is developed and looks like it might have a good signal, we have a duty of care to make sure that that treatment gets to everyone in the world. And that includes people in Turkey, in Iran, in the Middle East, in South America, in all the countries in South America. And it's something that I feel absolutely passionate about. We have to develop drugs that are accessible to everyone. So I think that industry partners who work on Huntington's also feel that.
And then for the last, it says, greetings from Turkey. Oh, no, that's not the last one. Somebody else has come in with another one afterwards. The first part of the question, I'll read it, but I'm not, when can we deliver the medicines to patients in Turkey? And then when you say young adult, especially, how old is the age based on?
So again, delivering the drug in Turkey will be for future trials, if there are sites suitable to do that with MRI and lumbar punctures, and I'm sure there are, and also the age in the new study is based on that cutoff of 48. So the post hoc analysis looked at people under the age 48 and under. In the new study, it is looking at younger patients, and the cutoff in the new study is similar. And that's the reason, because it's looking to see if the post hoc analysis stands up in a new study with younger patients and it's safe and tolerated. So that's where the age cutoff comes. Peter, did you want to add anything on that?
Just to reiterate what I said before, that age cutoff doesn't mean that in future studies we'd hopefully expand that, so we'd be able to treat people of a wider range. But again, we need to replicate what we've seen in the subgroup in Generation HD2.
And I'm going to go back to, it's more of a comment than a question. A new biomarker, IL-6, is a very promising on showing data for blood plasma on the early HD early stages. I hope it proves to be useful.
Yeah, we published IL-6 in plasma in 2008, actually, in Journal of American Experimental Medicine as signs of early inflammation. And we did a lot of work on trying to work it up as a biomarker for trials. IL-6 in plasma, it may end up being useful, but we don't know yet. But what does look like it's useful, which is an inflammation marker, is YKL-40. Now, YKL-40 is a brain inflammation marker, and we know in CSF that goes up very early in the disease. It goes up as early in the disease as neurofilament in stage 0 patients from our HD young adult study. So for me, measuring YKL-40 is important because it's a marker of inflammation. We know inflammation plays a role in the disease, but also it can be used to look and see if a drug is causing inflammation. So I think CSF YKL-40 as a marker of inflammation is also important. And that's being measured in the new study and in many different other clinical trials. So it's related to IL-6. It's just brain specific.
And the last 3 comments on here are just thank yous. Vlado, in terms of your comments, Sarah, about having access to medicines worldwide once there is a medicine. And just also another one, thanks, Dr. Sarah. And thank you, Dr. Sarah and Roche. You provide hope for us suffering. Sometimes this is all we have. Oh. And another thank you.
So just to finish in the last minute or so, I absolutely promise you we are passionate about trying to find treatments for Huntington's disease. And I can speak for that in myself independently. And we will keep striving until we have something that slows disease progression. And then when that time comes, we will make sure that it's able to be given to everyone worldwide. And I think that's something that all of us, patients, families, and doctors who work with you all feel very strongly about. So thank you for involving yourself in our studies. Patients are the solution. We can't find treatments for Huntington's disease without you all. So your voluntary participation in our studies is what helps us do the science and find treatment. So we can't do it. We can't find treatments without you all. So thank you.
Thank you. And again, thank you all for being here and posting your comments and questions and bringing things up to us. It's been really, really interesting and encouraging. You sound really kind of, enthusiastic, Sarah, which is a nice thing to witness and to have people like you and Peter and the others on board working to have treatments being possible in the future. So thank you so much. And I think also that what we have showed here is that we are a global community, and that we need to make more use of that global community. That's something we want to do whatever we can to make happen, and in the future. And of course,
Absolutely.
we need to take effort also to have future treatments available all over the globe. So with that, I just want to thank you again, and take care, and keep on.
Thank you,
Good
and
work.
Happy New Year.
Yes,
And
Happy New Year.
hope for 2024. Lovely to see you all.
Likewise.
Bye.
Bye.
Bye.